you guys can do some fun activity, right? All right, so last week we talked about physics of vision. This week we're going to talk a little bit about visual physiology. We understand that, like, what light is, waves, particles, all that kind of business. That's fun. So that's the stimulus, but we really need to think about that sensory apparatus for a little bit, right? We talked a little bit about this when we talked last week, but we want to add a few things this week. I'll let you know what's important because there's going to be some stuff we can just sort of breeze through, right? Here is you if the top of your head was cut off, right? And we're looking down. I know, right? It's very pleasant. So we're looking down at the head, right? Here's your left visual field. That's anything on the, let's just say, the left side, right? You've got your right visual field. Guess where that's located? It's on the right side. Down the middle, which you can kind of figure out where that is, most folks have a nose right there. We'll call that, because it's going like up and down, right? We'll call that the vertical meridian, because it's like vertical and it's in the middle. That's pretty cool, right? If we were looking at this from the sides, we take that and kind of rotate a little bit, and coming out from your eye, like straight out from your eye, that's another line we'll call the horizontal meridian. Anything above that is going to be upper visual field. Anything below that is lower visual field, right? So we've sort of created pretty quickly there 4 quadrants, right? We've got an upper left, a lower left, an upper right, and a lower right. That's pretty awesome, right? Okay. What's interesting about this is something we call the partial decussation. It's a really big word to mean some stuff crosses over and some stuff doesn't. Okay. How many of you know just kind of that basic, well, what's in your left eye goes to the right side of your brain, what's in your right eye goes to the left side of your brain? That's almost true, right? I mean, it's true for some information, but not for everything, right? Because if you close one of your eyes, you can still see things over in the other hemisphere, or other in the other visual field, right? So if you just leave your right eye open, you're not only seeing right visual field, you're also seeing a little bit in the left visual field. Okay, and what's important is left visual field crosses over, right visual field crosses over. If you're not able to see things on either side, Eli, you need to go see a neurologist because something's wrong with your face. Because your nose is shifting. Right? You don't get much, but you get just a little bit, right? And that's because humans, unlike other species, how many of you know about mules? Yeah, mules are awesome, right? You know the cool thing about mules? There's a lot of cool things, Laura, just hang in there. But the main thing we're concerned about with it, why do they call mules sure-footed? Anybody know? They never say a horse is sure-footed, right? You think, well...
They don't fall, right?
Right.
Yeah.
But what's interesting about a mule is the way their heads are shaped and their eyes are located. You can actually see their back feet while they're walking.
Ew.
I know, right? Isn't that the coolest thing ever? You can, I mean, they can see their front and back legs.
I know, that's weird.
It's kind of weird, isn't it? So they can see where they're putting their feet. So that's why they're sure-footed. They don't have a lot of what we call binocular vision. Okay, there's not a lot of overlap because their eyes are kind of out on the side, right? You guys know what a horse head looks like, right?
Yes.
Okay, just giving people a moment because not all of you are equinologists. Is that what you call someone who studies horses? Probably not.
You could. Don't know if you'd be right. But
Close enough, right? So not all of you are horse people. I think that's centaurs. That's something different too. So, horses, they have their eyes on the outside. They don't have a lot of binocular vision. Humans, our eyes are forward-facing, right? So both of our eyes look forward. There's some region in the middle there that's binocular. So if you close either eye, there's a good bit of space in the middle that is covered by both eyes, right? That's the binocular area. It's where we get that nice depth perception. We'll talk about that in a few weeks. That's why it's important to think about this as a partial crossover, because what's in the left visual field, whether that's hitting your left eye or your right eye, that goes to your right hemisphere. What's in the right visual field, again, whether it's coming into left eye or right eye, doesn't matter. It's going to your left hemisphere, right? Does that make sense? So some of that's already on that side, right? So some of what you're receiving in your right eye is already coming from the left visual field, so it doesn't need to cross over, right? It's already on the correct side of your brain. Does that make sense? Once we do that little crossover, we go a couple places. There's some information that goes to this guy called the superior colliculus. Colliculi is plural for colliculus. It means little hill. So some information goes there, coordinates eye movements, integration with hearing. We'll talk about that when we get to some sensory integration. The bulk of this info goes to an area called the lateral geniculate nucleus. Specifically, this is the dorsal lateral geniculate. There's also a medial, which we talked about when we talked about hearing, right? The medial geniculate. But we want to think about the dorsal lateral geniculate. We're going to do some interesting processing there, and then on back to visual cortex. Some places. There's a lot on one slide, right? We've already talked a little bit about the retina. We talked about photoreceptors last week, rods and cones, right? We said rods are more sensitive. They actually have a slow response, though, right? So they're more sensitive. That's why you use your rods at night. You can't see things, remember, in the middle, because you're not fixating on that, right? You're just getting kind of information from the periphery. You think you see things when you look at it. This is not to scale, and I hate when they do this. Rods are actually much, there's a much different sort of size between that and cones, but they drop off to the same size, doesn't matter. These cones, why are they called cones? Well, they're shaped like a cone, and shaped like a rod. Not a big deal. These little stacks is where the photopigment molecules are. So when a photon of light comes in, right, it really needs to hit a molecule in one of these stacks. It's going to take that molecule and it's going to change its shape. Don't worry about this too much. It's going to change its shape, and that shape change causes a cascade of other events that say, hey, we absorbed a photon of light, and then we will change the output of that photoreceptor accordingly. Whether or not a photon gets absorbed is really dependent on three things. One is going to be direction, right? If the photon is traveling sort of through the center of the lens and perpendicular to that stack of photomolecules, it's going to be more likely to get absorbed, right? So if you go back here and you think about something coming in directly straight, look at all of the opportunities for it to hit a photomolecule, every one of those stacks, right? If we come in at an angle, just that little bit of an area there where we could possibly hit a photoreceptor, so it reduces the likelihood that we'll absorb that photon and recognize that that light is in our eye, right? The next thing is frequency. Each receptor has a certain sensitivity for frequency, right? We talked about frequency some last week. Each photoreceptor is sort of grouped into 3, 4 if you count rods, spectral sensitivity ranges, right? If that photon of light, if that frequency is right at that peak, if that frequency is right at the peak sensitivity, that photon is going to be more likely to absorb it. As you get away from the peak on either side, that particular type of photoreceptor is less likely to absorb that photon. It can still happen, right? Sometimes things sneak in, but not all the time. Finally, the thing we want to think about is once a photon's been absorbed, you have no way of knowing what the frequency was. Okay? We have 3 types of cones: long, medium, and short wavelength sensitive cones, right? If a medium wavelength cone accidentally absorbs a short or a long wavelength photon, it's going to be perceived as a medium wavelength, right? Because that photon is what received it, okay? Does that make sense? No? Have you ever had one of those, like, one of those banks? They used to make these a lot. You'd put just like pour your change in, and then they're like self-sorting, right? So like all the quarters, nickels, dimes, pennies, they all fall in. And then however far that stack is up, like on the outside there's a scale and it'll tell you like how much money you have, right? So what if you're doing that and accidentally a penny falls into the quarter slot? Well, when you look and you read that output, you're going to read it as extra quarters, right? So you have no way to know that that was really a penny. You're just going to measure it as a quarter because it fell into the quarter slot accidentally, right? So if that photon falls into a medium wavelength photoreceptor, you're going to perceive it as a medium wavelength, even if it was a long or a short wavelength. Is that cool?
How does that happen in real life though? Like what, like?
It constantly happens in real life. It doesn't happen often enough to give you a problem.
Is this like, I guess my question is, are we like cognizant of this happening?
No. I mean, it happens for most of the time. It happens at such a low rate. It's kind of like that random noise we've talked about before, right? It just gets sort of factored out. The other thing to consider is everybody's, there's some variation in the way these photopigments are structured, right? And the way your photoreceptors are structured. And we'll talk about color blindness at some other point. This will make a lot more sense at that time, right? And so everybody, your photoreceptors, some are more sensitive to different wavelengths than others, right? So if we were to look across individuals, let's say somebody had a color vision deficit, right? Let's say they weren't color blind completely, they weren't missing a total group of photoreceptors, but one of their photoreceptors was shifted a little bit, right? They may be more likely to absorb photons at a different wavelength. But it just happens accidentally too, right? Basically, the way I like to look at your biological processes is anything that's ever going to happen in your body is happening in it right now. It just might not be happening at a rate that matters, right? So you are going to accidentally be breaking down enzymes that you need, right? And you're accidentally going to be making enzymes that you don't need, right? But it's are you making enough or breaking down enough to cause a problem, okay? And typically you don't. And when you get out of the range there, that's when you cause some pathological problems. Don't think there's any way you could force this to happen, right? Like accidentally. Just trying to think about ways that you could do it. I don't think you can. You'd have to really alter the structure of your photoreceptors to do that, and the photopigments inside. Does that make sense?
Yeah.
Is it like when that happens, it's like, do we either process and like see it, or like is it just?
Probably not, because so you don't have to take much from this diagram, right? But this is actually a really good sort of segue into something called lateral inhibition. Okay, so when you have these rods and cones, you have these horizontal cells in between, and what these horizontal cells do is they help sort of factor out accidental responses from these neighboring cones, right? So let's say that I want to ask everybody in this class, hey, did you hear something, or did you smell something, did you see something, right? And if I were to ask you, do you, is that, and you say, yeah, I smell something, it smells like something's on fire. Well, I'm going to ask the people around you, right, if they also smell like something's on fire. And if they all say, no, I think, Ree's having a stroke, right, which is a possibility, then I'm just going to ignore that, right? But if like 3 of the 5 people around you also say, yeah, like I smell like something smells like it's on fire, right? Then I'm going to say, well, there's probably something there, right? So if that like aberrant accidental photon comes through and you go, whoa, I see red, and then everybody around you is like, no, we don't see red at all, then that lateral inhibition is going to go, shut up, you're wrong. Does that work?
You're gonna ignore if a student has a stroke?
It's not part of the FAST acronym, so.
Yeah, it's not, is it? What is part of the FAST? Rub your face. I don't know what the A is. Some of the face.
Arms. It's face drooping, arm weakness, like slurred speech, and time. So you have like half an hour to get to the hospital.
Hey, at least you're on wheels. Just push you down the stairs. That'll get you there fast, right? Who's got a phone? They call 911. I'll just push you down the stairs. We'll keep going.
Why don't you put it in the elevator? Why don't you push it down the stairs?
Have you been on that elevator? It takes forever. Time.
Then I think she'd be nervous.
If you're having a stroke, don't worry about a broken arm. I mean, that'll heal. Your arm heals faster than your brain. That's a fact. It's on Wikipedia.
Considering how much people fact check Wikipedia, it's probably not too inaccurate.
It's probably not too inaccurate, yeah. So there you go. It's good advice, right? Are you all right? I probably wouldn't push you down the stairs, just like maybe to the edge. And then if let you make the decision about going on down on your own.
So it's not his responsibility if you don't.
Yeah, I'll just set you up put you in the fork in the road. You pick which way you go. Yeah, stuff to come.
Tumble down the whole flight of stairs.
let's talk about on versus off bipolar cells. So when we go back to this, we had the cones, right? We have rods. We're not going to worry too much about rods. So we kind of just ignore this half the diagram for now. You've got cones. We've already talked a little bit about horizontal cells, right, and that lateral inhibition business. So we kind of think about that. The next thing down is we have what are called bipolar cells. Okay. These guys take information from cones, and they'll eventually send it to things called retinal ganglion cells. Now, these retinal ganglion cells are important. They're the guys who actually send the big signal to the rest of your brain that says, yes, there was light. Okay? So the retinal ganglion cells don't fire an action potential. They don't send that signal. You don't perceive anything. Right? So these bipolar cells, some of them are what we call off bipolars, and some are what we call on bipolars. So what does that mean? That basically means if you're an on bipolar, right, and a photon hits you, or hits the photoreceptor that you're getting information from, you get excited about that, right? That receptor caught a photon, it said, hey, I caught a photon, you're like, great job, I'm excited about that. That lets me know I saw something, okay? The other bipolars are what we call off bipolars. So whenever that photon hits that photoreceptor, and then that photoreceptor goes, I caught a photon, that bipolar goes, I'm off, right? Because we also want to know, we kind of want to have that sharpened response, right? So we want to have ons and offs, right? And these guys are going to overlap, so we're constantly turning things on and off, right? And so we know where activity is and we know where activity is not, right? That's why we have ons and offs. It's sort of like, trying to think of a good example. It's like the temperature in your house, right? Because you want to know if it gets too hot or if it gets too cold, right? You don't just want to know if it gets too hot and things can get cold and your pipes can freeze, right? You also don't want to know if it just gets too cold because then it can get too hot and like your cats can catch on fire. Right? I don't know, like spontaneous combustion of felines. They're already like stirring up that dry litter anyway. Cats are a fire hazard.
I hope
You have a cat?
I have 2 cats.
Fire hazards, aren't they?
Yeah, one of them was like so stressed that she was pulling out her fur, so even more of a fire hazard.
You can, so my sister-in-law has a cat that's sort of anxious all the time. She gives it CBD oil. Seriously, that's what her vet said to do.
Oh my god.
No.
My vet prescribed us like an anxiety medicine, and then
Yeah.
a week later we found them all underneath the bed where she would go and like spit them out after maybe 2 hours.
I don't think it would do that with CBD oil. You should give that a try. See, I should have been a vet. All right, so cones, what do we need to know? We already talked about on and off cone. Hey, horizontal cells, great job asking about that before the slide came up, that's great. Amacrine cells do something similar. They're just between bipolars and ganglion cells. Now we need to talk about ganglion cells. These guys are important, right? These are the major output from your retina. It says there are 4 major types. In fact, there are like 20 types of ganglion cells. And they constantly keep adding new types of ganglion cells. I am not going to expect you to know all 20 types. Probably not even going to really expect you to know these 4. We're going to know some of them because we're going to talk about them a little bit. So no worries there. You've got your biplexiform, your bistratified, your midget, and your parasol. Right? And as it says here, who knows what those biplexiform do? A lot of these are actually involved in diurnal rhythms. So there are, for those of you interested, there are retinal ganglion cells that are sensitive to light. So they're sensitive, they're like photosensitive. These project directly to your hypothalamus, which is the guy that tells you, hey, it's time to get out of bed, lazy, the sun's up. I don't know if any of you have ever tried to sleep, you know, when the sun's coming through your bedroom window. It's kind of difficult, right? Because that light comes in your eye, triggers a bunch of hormones going on, and you're supposed to be awake. So there you go. But we will talk about some of these other guys as we All right. So the midget cells, right, they are, in fact, the vast majority of the retinal ganglion cells that you have, right, are going to fall in this category. Really high light, that's what we mean by photopic. It's going to need a lot of light. So based on that information, you should already know that these guys are collecting information from cones primarily, right, because cones require that high light level. They will project to something called the parvocellular layers of that LGN that we talked about a few moments ago. Anybody know what the word parvo means? Not that disease. Parvo means small, right? And so that kind of falls in line there with your midget cells and the parvo, right? The other type of cell we'll talk about primarily as ganglion cells are going to be parasol cells. Not as many of these. They have some photopic light, but also scotopic, which is kind of those low light levels, so they're going to get some cone information as well. These project to the magnocellular layers. Anybody know what magno means? Yeah, if parvo means small, magno probably means large, right? Those third guys we talked about, the bistratifieds, they're kind of interesting. They probably handle like blue light. Just kind of interesting that blue light's separate. That'll make some sense later when we talk about color vision. Your what we call red or green cones, which we'll fix because you shouldn't call them that. Those are long and medium wavelength sensitive cones. They're very similar genetically. That's why red-green color blindness is the most common, right? Because if you have a small tweak in either of those cone types, it brings their spectral sensitivity so close together you cannot tell the two colors apart. Whereas your shortwave cones are kind of way over here, right? And they're handling blue light. And that's going to project to the coniocellular layers of the LGN. Anybody know what conio means? If parvo meant small and large meant, or magno meant large, then conio must mean dust. Means dust.
Okay, why would you do that to us?
Yeah, well, just because.
You should have been a vet.
When these cells were first discovered, people thought they were useless. They're like, oh, look at those dust cells. They're wasteful. And then later on, folks actually found like, well, sort of in fact, they do something important. But after that, they'd already been named useless. So we're not going to change it at this point.
That literally translates to useless?
Dust. It's dust.
Oh.
I mean, dust is useless, right?
I mean, yeah.
Nobody says, boy, I wish I had a big pile of dust to accomplish this task. I don't think anybody's ever said that.
So they require a lot of light as well?
They do. They're photopic, so they're getting stuff from cones. They're getting L and M, so that's long and medium, and they're comparing that to the short wavelength. So that's like short versus long and medium added together, right? So they're comparing that. Where these guys over here, they're comparing like long versus medium, or just looking at long and medium put together. Does that make sense?
I'm lost on the dust part.
On which part?
The dust part.
Don't worry about it.
Okay,
That was the least important part of the whole story.
alright.
So here we go, when we're looking at, and this is going to be important for a couple weeks from now, because in a couple weeks you guys are going to plot receptive fields. It's going to be fun. It's a brilliant lab activity. I know, guess graph paper, all kinds of graph paper, and numbers and charts. And if you have colored pencils, I would bring those. Anybody have colored pencils? So I don't know if this is next week or 2 weeks from now. Start bringing colored pencils with you every week. Does that seem reasonable?
I have no idea what you just said.
Bring colored pencils all the time. So here is an on-center ganglion cell. If the center, guess where the center is located? That's this area right here. And then we have one other area. This is going to be really complicated. We gave it a really, really difficult name, Emily. We called it the surround. If you've got the center, then you've got what's around that, which is the surround, right? That's this sort of ring. Real technical name for that is an annulus, right? Anybody into geometry?
It's my least favorite one.
It's the best though, makes all kinds of pictures. So the annulus is just a ring, right? Why would we call that an annulus? If something is an annual, it just goes around every year, right? So it's in a circle. So we have the annulus. That's pretty good. If light falls into the center, look at that. That cell gets really excited, right? Because it is an on-center cell. Now, if it's an on-center, then the surround is off, right? Because it's the center surround opponent processing, right? So if the center is on, the surround will be off. If the center is off, then what's the surround? Yeah, right? So it's just the opposite. So here you go. If you excite both the center and surround, you get a few little responses, not quite as much as up here, but more than nothing. And if you excite, or just, just put a stimulus onto the surround, then you get this massive inhibition. When you release that, you get a little bit of rebound, right? Because you've been holding back, Gabby, all of those spontaneous action potentials, and then when you release that inhibition, then you get a few of those right together, right? So you're going to have that kind of brief pulse of action potentials. It's just called a rebound. Does that make sense? It's kind of like, I don't know, anybody have a dog? Yeah. And so if you take your dog and you tape it to the floor,
Never done that.
and like hold it still for a long time, and then it's kind of like really wants to be active, right? And then if you like cut the tape and you let it go, then it's going to be overactive for a brief period of time.
There are a lot of questions that we can unpack there, but
Maybe you shouldn't have been a vet.
So my sister, when she was in high school, worked for a vet for a while, right? She was like, I don't know, kennel master or something, I don't know. She did not, but one time, this is the craziest story, she helped hold a hamster still. It took 3 people. They put a cast on its leg.
Oh, that's so
Yeah.
cute.
that's so pointless.
That's what I thought. It's like, how much did they pay to put a cast on a hamster?
Just get a new hamster.
Maybe they really love that one
God.
particular hamster.
It's a
But what's the
hamster.
lifestyle, I mean, what's the lifespan of a hamster? Like 6 weeks?
3 to 5 days.
2 weeks? Is that
It's 2 years.
2 years? By the time you put the cast on it and that leg heals up, it's dead.
Exactly.
God.
Thank you.
Would you
be so insensitive?
It's a hamster, okay? It's a rodent. It's basically
take
a
a
rat.
popsicle
like,
stick
it's
to it and go?
That's a stint.
But they put a cast on this hamster's leg. I don't even know how they, and I don't know why it took like 3 people to hold it.
How big was this hamster?
I'm like, I got a hand, it's still,
Those things freaking hurt though when they bite.
I would have anesthetized it. Give it a little bit of ketamine and you're done.
Oh my God.
By the way, ketamine is an actual veterinary anesthetic. If you've ever taken your dog or cat to the vet and it's been anesthetized, I'm quite certain it had ketamine. Okay? That's not me just saying like give it a weird drug because you guys don't know the actual purpose of ketamine. Right? You guys are spraying it on walls and licking it off to try to have hallucinations.
Do you hug animals? Do you ever hug them? Do you have one? I'm worried about you.
I don't think you have.
Do you have one?
There's some cats.
No,
at my
you
house.
need like a
These are
dog.
fire hazards.
Yeah.
It's personal
I
experience.
have to keep paying my insurance because insurance went up because I have 2 cats at home. Fire risk.
They're just a walking box of tinder.
They are. My cat almost sent an email the other day. I left my laptop open. It jumped up and it had a box up with like.
It was gonna
Stuff
tell
in-
on you. That's what was gonna happen. Please help me. Off-center ganglion cells are the exact opposite, right? If light falls on the center, we don't get a response. If light falls on the surround, the cell gets activated or excited. It's not a big deal. This is what you're going to have to do in a couple weeks. That looks like nothing.
This is easy. It looks complicated. It's going to make a lot more sense later. We'll briefly talk about it now, and then the lab activity is going to be cool. This is basically a cross-section, right? And so what you've done is if you take this and you sort of, it's like this, and if you turn it on its side, what you'll see is sort of 2 circles. And you can make a heat map, right? So basically this is the size of that receptive field in minutes, because we have like degrees, but then you go, if it's less than a degree, it's minutes, right? Okay. So this is like, 200 minutes, right? Negative 100 to 100. And if we're just looking at that center part, like the excitable region, then we can plot that, right, with this blue line.
You'd say, oh, it gets really excited in the middle, and it kind of falls off as we go out to the edges of that receptive field. Then if we look at the inhibitory surround, and we can kind of plot that, right? That's kind of cool. Then what we can do is this thing called a difference of Gaussians. These are cool dogs. It's a DOG. They won't cause you any problems. Difference of Gaussian. You can subtract that that inhibitory portion from the excitatory portion. What you're left with is going to be that receptive field. And so what you're left with is something a little bit less excitable, dips down a little bit. And when you look at the structure of that receptive field, if you think about anything that falls above this line, is going to be the excitable part. That's the excitable center, right? Anything that falls below that line after this subtraction is going to be that annulus, that surround, right? It's pretty cool. It's kind of like a sombrero, and with the shape on the side, right? Because you have those kind of dips, and then you have the peak of the hat. You see it, right? It makes a lot of sense. And so if you take the brim of the hat and cut across, anything that's above that is going to be the excitable region. Anything below that, the dip in the brim, that's which goes all the way around, is going to be that inhibitory surround. Or if you flipped it upside down because it was an off cell. Don't worry about that. Does that make sense to everybody?
And you're supposed to graph this?
Next week. Don't worry about it now. So this is a midget receptive field, and then here's a parasol receptive field. And so what do you notice that's different between the two of these?
Parasol's wider.
Yeah, the width, right? So the parasol cell, it's wider. Why would we want most of the cells in our visual fields to be these, or in our eyes to be these midget cells? They're smaller, right? And we were sampling from smaller little pieces of the world, and we get a more accurate and resolved picture, right? So basically, this is like 4K, right? And this down here is like 720p. Is that terms you guys understand? You guys have televisions,
Or
right?
like pointillism.
Huh?
Pointillism.
Pointillism is very similar, right? So when you get up to pointillism, it just looks like a bunch of dots, right? But when you get far enough away so that those dots and your receptive fields kind of reach that nice sort of point where it's all, you can't tell the difference between the dots, it makes a nice picture, right? You guys like pointillism? Who's an art minor?
Major,
Major?
yes.
Why are you in this class?
Because I'm dope majoring.
Actually, this is the best class you will take for your art career.
It's cool so far, yeah.
Just wait, because now we've started vision. What we're going to be doing from now on, it's going to be, you're going to go, wow, this makes so much sense now. What media?
What medium?
Yeah.
All of it. But I've done pointillism before. It's cool. It just takes a lot of time.
Yeah. It's a lot easier to just look at it.
Yeah.
Let somebody else do the hard work. The spatial and temporal responses, I'm not going to ask you too much about this. All right. Retina, we've already talked about this, right? A million ganglion cells. Okay. Now there are 120 million photoreceptors, right? So this brings up the concept of convergence. Okay. I know it's pretty cool, right? So we've got 120 cells funneling down to 120 million cells funneling down to 1 million cells, right? So it's 120 to 1 ratio. Now, most of that convergence is occurring in the periphery, right? Not in the retina, or not in the fovea of the retina, because that's going to be mostly one-to-one relationships. Okay. All right. Don't worry about this too much, because we will talk about color vision later and spatial vision at a later time as well. So those are separate chapters going down the road you guys are going to enjoy. All right, so where are these cells going once they, or this information going once it leaves your eye? Most of it goes to the lateral geniculate nucleus, almost 90% of the output of your retina goes there, so that's pretty exciting. Some of it goes to the superior colliculus, we talked about that. Suprachiasmatic nucleus, we talked about that. We've got some other areas as well. We're not going to worry about that too much, pupil diameter, things like that. Those are important. We're going to do some pupil tests today. You're going to get to shoot lasers in people's eyes. So be excited. No, I'm just kidding. We are going to, though, watch the pupillary response, which is going to be exciting. We already talked about partial decussation and that LGN business, right? Here is your lateral geniculate nucleus. This is a terribly complicated thing that I'm not going to expect you to know. Okay? So don't get too wrapped up about this. The LGN has 6 layers. You need to know that. That's what this guy looks like here, right? 6 layers. Geniculate, by the way, that means knee, right? So see how it's bent like a knee? I know, right? You guys can see that. Some of these layers are from your, what we call the contralateral eye. That's the opposite eye. Some of them are from the ipsilateral eye. That's the same side eye. Again, Tabby, that's because not everything crosses over, right? So some's coming from the other side, some's coming from this side. Some of this information, you can see these 6 layers here. Some of them are magnocellular layers. You got a couple of those. And then you have 4 layers that are parvocellular. Why would we have more layers for parvocellular? They're smaller receptive fields. We have more of them, so we need more layers to process that information, right? It makes perfect sense. And then you've got those layers in between those coniocellular layers. Now, there's an interesting pattern how this comes in, but don't worry about it. I've already talked center surround. Not a big deal. The LGN does get information that's not coming from your eye, right? Some of it is coming from visual cortex back down. Just as when we had in the auditory system, we had those outer hair cells, remember, that were doing some fine tuning of that response, like saying, hey, here's the information that's been coming in, let's predict a little bit about what's happening so you get ready. The same idea is happening in the visual system, right? We call this a feedback projection. So information that's coming back down in the opposite direction to help tune those responses. The other information that's going in the normal direction, we call that feed forward. Once we leave the LGN, we move up to visual cortex. There's a complicated pattern there as well with your 6 plus or minus layers in visual cortex. I'm not going to expect you, JC, to remember what each of these levels is doing, but just know that some of the levels are levels that receive information, and some of them are levels that send out information to other places. Okay? So you've got some input layers, some output layers, you've got some layers in between that are doing some internal processing. That's really what cortex is doing just about everywhere. Okay? If you guys are really interested in, geniculo-cortical connections, feel free to really get excited about that. We can talk about it. It's a lot. There's a lot of information, a lot of cool stuff going on there. All right, moving right along. What about orientation tuning? So previously we talked about all receptive fields being sort of donuts, right? Centers and surrounds. Once we get out of the lateral geniculate nucleus, we go to visual cortex. We're taking a bunch of donuts, and so what's better than one donut is a whole box of donuts, right? You guys love boxes of donuts? No, not a donut fan?
Donuts are great.
I know, I think somebody's failing this class. How can you not like donuts? I mean, there are people who are like, well, I shouldn't eat donuts.
Yeah, that's more
that's
of what
different.
it is.
That's different.
It's not that I don't like them, it's just what do they do for you?
Provide happiness.
Nothing.
Release dopamine.
Release dopamine. Yeah, right there.
Again, provide happiness.
Both of you were right on, somebody gave a more technical answer, someone gave a more qualitative answer, that's fine. Both of us were right. Huh?
You go for a run, it does the same.
Yeah, but it's small.
See, that's the face I made. Laura's like,
Running just makes me sad, so.
you should run with it. See,
Sonny's
look,
got
someone
the whole
has
thing.
donuts. So, Sonny, can I have 6 of those? No, I'm just kidding. Just kidding. No, she doesn't have them. Do you want to hold up a donut and show
I
everyone?
already ate half of it.
It's okay.
I'm pretty sure everybody knows what a donut looks like.
It's fine. So, in visual cortex, we're taking those center surround receptive fields, Mary, and we just take them and we stack them next to each other. Oops. That didn't really do what I wanted to. Oops. And so you can kind of see in this phenomenal, you can tell I'm not an art major.
You're doing great.
I know. And this is with the mouse on my odd hand, too. So normally I have the pen in my actual like hand that I use for drawing and writing. This mouse is over here. So if we take one of these center surround and we stack a bunch of them together, what you now have is sort of all of these, let's say, excitatory centers, right, lined up. So those are really powerful. So you have this nice strip of that. And then you sort of have these inhibitory flanks, right? See how these areas sort of overlap? And they create these inhibitory flanks. And what you end up with is a structure like this, with an excitatory center and two inhibitory flanking regions. We call that a simple cell. It's a pretty simple structure, right? So now, instead of dots of light, which are fine, we've taken those dots and we've made them into bars. And I know right now you're thinking like, why do I care about bars? You care about bars because you walk out of doors, right? So if you think about this door, and this door is made up of a series of bars, right? And if you could only see dots, and you're not able to integrate those dots into a larger structure, and you just see the whole world as a bunch of little dots of light, you're not making any sense of that, right? You have to take those dots and put them into groups. This is the first stage of that. Later we're going to talk about more complicated shape and object recognition. This is the first foundational step of that process. That's pretty cool, right? Now there are some cells that are wired up differently, where they've just decided to pluck from the excitatory centers, and we'll do that and we can create these sort of complex cells that don't have inhibitory regions. Don't worry about that too much, just know that's the thing that happens. Is that good? We'll talk about direction selectivity later when we talk about motion. So again, don't worry too much about this right now. Worry about it later. Oh, who loves ocular dominance? Nobody? Don't know what it is. But I'll tell you. If you take the cells in your visual cortex, right, and you say, hey, is that cell getting excited from information from the left eye, or is it getting excited by information from the right eye, or does it get equally excited from information coming from both eyes? That's what we mean by ocular dominance. Most of the cells in your visual cortex are driven by either the left eye or the right eye. There are a few cells that would kind of fall in this middle region that are equally excited by the left and the right eye, but most of them prefer one eye or the other. Okay. These guys are important, Hubel and Wiesel. Maybe in a couple weeks we'll watch that video. Kaylin, that's a great video, isn't it?
They're thinking about it, still haunts me.
Really? You didn't even get it. I mean, the cat's not even in the video.
I know, but it's just the idea.
I guess.
It makes me sad.
It's a cat video. Guys won a Nobel Prize for recording responses from the visual cortex of a cat. It's pretty cool.
They had to kill it afterwards.
Why?
That's
Because
how
they had to
science
poke
works.
its brain to see the responses.
Was there not a rat available?
The problem with rats.
There was hamsters.
The problem with rats is rats are primarily olfactory. You know, that's how, and somatosensory, that's how they get around. They don't really get around with vision,
Sure.
right?
Fair enough.
Cats are, you know, they have more forward-facing eyes, so if you want to look at like binocular kind of issues, cats are better for that. If you want to do a few other things, cats have better visual systems in general. What you need to also do once you've done an experiment like that, the reason you have to verify where you were in the cat's brain, right? What if you were recording from auditory cortex and you got these like weird visual responses, right? So you have to verify that. So eventually you have to remove the brain and examine that tissue. We talked about this when we talked about methods. Weren't you guys paying attention then?
Probably not.
In fact, far fewer animals are euthanized in scientific studies each year than are euthanized at shelters. So if you really want to have a major impact on the death of animals, you should snip every animal you find.
Just myself. Just go ahead.
Hey, people are animals.
Handle it.
So,
People are animals.
true story.
We're
gotta-
snipping people too.
There are too many of you. I mean,
Actually, there's not enough according to the Census Bureau.
I mean,
Well, the
We're
Census
not-
Bureau is an idiot.
I'm just saying.
And I mean that in,
We're not having enough to replace the population.
right, which is good.
For you, yeah, you feel that way.
Well, I mean, the planet feels that way.
Sure.
Because it's really trying to just shrug as many of us off as possible. Sooner it does it, the better.
Are you gonna be the first one to volunteer?
There's this whole group of folks who are like, what's the name of this group? For the like, people for the voluntary extinction of humanity or something.
That's a thing.
You started it, didn't you?
It's a
Google
thing.
it.
I did not. I'm afraid to type in because the acronym is like very similar to some things I shouldn't type for some reason. I don't remember what the acronym was, but it was like something like, if I misspell that, I think something's coming up that I don't want to show you.
I'll find it.
3 scrolls, 3 images.
3 scrolls.
There you go.
But it's folks who've decided like, we're not having children, we're going to be the last of the human race because, you know, we're sort of screwing things up. Now,
so like every day.
there you go. Right?
There you
Thank you
go.
for saving the world, you're welcome.
They are. They're just taking in other people's kids that don't take care of them,
Yeah.
right? Mason?
VHEMT,
VHEMT.
Voluntary Human Extinction Movement.
Yeah, that's it. They'll send you a sticker.
A t-shirt?
You guys are into stickers?
It'll be fine.
Not for that cause. Not for that cause.
Only if it's a star.
Only if it's a...
Gold stars or nothing.
If I'm gonna sacrifice myself for the good of the human race, I better get a fucking gold star.
We're not sacrificing you, just half copies of you. Don't worry about it. Hey, who remembers that time we talked about cortical magnification? Here it is again. Again, the central little smidge of your vision is the most important part, so it gets the biggest part of your visual cortex, right? That's all you need to know. Ooh, hypercolumns. These are fun, but we're not going to talk about them. It's going to blow your mind. But hypercolumns are super cool. It's the way visual cortex is organized so that like every chunk of cortex contains all of the cells necessary to process any sort of orientation or contrast that you would experience in that particular spot of the visual field, right? It was this idea kind of put forward by Russell and Karen DeValois. They wrote this great book called Spatial Vision. If you guys got really excited about Fourier transforms, Sonny, write that down, because I know you did. That's a book you should read.
Did you read it?
I have a copy in my office.
Did you like it?
I did, I enjoyed
But
it.
did you read it?
I did
Or is
read
it a paperweight?
No, I've read it 2 or 3 times. JC, you got a question?
Can you explain those one more time?
So hypercolumns is basically if you take a chunk of cortex, right? Just like a cube of cortex, and suck that out. There are going to be cells in that cube, right, that are all responsible for one little dot of the visual field. But all of those cells some of those are going to be processing vertical bars or slightly, tilted bars, right? Like all of those possible orientations are going to be included in there. Probably also cells for each spatial frequency. We'll talk about spatial frequency another day. So like all of these cells are there, right? That can process any kind of stimulus that's in that particular part of the visual world. And that makes a lot of sense, because how many of you only look at vertical lines in the upper left quadrant? Nobody. You get vertical lines everywhere. So every single spot should be able to process vertical and horizontal lines, right? If not, then you're not able to see things. So that's something. All right. Once you get out of primary visual cortex, right? So you're coming up. You go to a bunch of other areas. We're not going to spend a lot of time on those today, because as we talk about each sort of visual capability later, we'll talk about the cortical area that's involved in that, right? Some of them are going to be involved in object recognition. Some of them are going to be in more location and movement, right? Visual motion. So we'll cover those as we go each way. Does that make sense? All right. The story I will tell you, and most folks will tell you, is that, area MT is only responsive to movement, right? That's not entirely true.
The major proportion of those cells in area MT are direction selective, right, which means they are sensitive to movement. But there are also cells in MT that are orientation selective only, or sensitive to color input, right? We're looking at what's the greatest proportion of the cells in that area, and that's going to tell us what that particular cortical region does, or the particular domain of vision that it's responsible for. Does that make sense? Okay. Who remembers the what and where pathways from the auditory system? Can we talk about that? Yeah, you've got the same idea here. You've got the what and where pathways with vision. You've got that dorsal stream, that's going to be your where. Ventral stream is going to be what. So these guys are going to do object recognition, V4 and IT, but like MT and MST are going to do visual motion. Okay. Again, we'll talk about these areas in detail later. There's some other ways you could divide this, right? Some people are like, oh, what about, magno versus parvo streams? Well, that basically shakes down to a where and a what. So really kind of looking at the same thing. And then there's action versus perception, which again is sort of still a what and where, right? So it really all kind of comes down to the same thing. So what and where is kind of the most common way that we think about dividing up your visual streams. All right, there you go. Right? That's not so bad. So any questions about that?